Plants lose their leaves and stems each year, and sometimes the entire plant dies. Ecologists refer to this dead plant material in ecosystems as litter. This litter contains complex organic compounds that are broken down into their simple inorganic constituents during the process known as decomposition. Decomposition has been well studied in grassland and forest ecosystems, but we're not able to use this information to predict decomposition rates in deserts. This suggests unique processes may be at work in ecosystems with hot, sunny, dry climates. This project tests the idea that light energy, particularly ultraviolet radiation, interacts with soil erosion to drive decomposition processes in deserts. To study decomposition, known amounts of plant material are placed in porous mesh bags. The bags prevent the litter from blowing away or being carried off by animals, but allows the litter to be exposed to light, water, and air, all of which influence rates of decomposition. These bags are collected over time and weighed, and the weight losses are an indicator of decomposition. We'll place these litter bags in areas with contrasting amounts of grass and shrub cover. Some litter bags will be exposed to full sunlight, others will be shielded from ultraviolet radiation. This will allow us to determine the importance of photodegradation, or light-induced degradation, in the decomposition process. Soil particles are carried by wind and water in the process of erosion. Vegetation structure affects the rate, magnitude, and pattern of soil loss and soil accumulation. By placing litter bags in areas with different levels of shrub and grass cover, we will be able to determine how vegetation influences soil accumulation on litter and thus influences rates of decomposition. Rates of soil movement will be measured in a variety of ways, including Wilson-Cook samplers, shown on the left, BS&E samplers at different heights, as shown in the center panel, and, as shown on the right-hand panel, by sensors that record the frequency of dust particle impacts. These samplers will be located in areas with different abundances of grasses and shrubs. Field studies will be complemented by controlled environment studies, where we will expose litter to different amounts of ultraviolet radiation and different amounts of soil cover, and then measure its mass loss. We also want to know what is the role of microbes versus light energy in the decomposition process. To address this question, we will determine what groups of bacteria and fungi inhabit the soils that accumulate on plant litter, and how their activities might influence rates of decomposition relative to photodegradation processes. Here you can see rhizobia on clover root hairs and ectomycorrhizae, two important groups of microbes in the soil. To identify these organisms, we will take advantage of the fact that microbial taxonomic groups produce signature lipids in their cellular membranes. These lipids can be extracted, identified, and quantified using phospholipid fatty acid analysis. We will use this information to determine what groups of bacteria and fungi inhabit the soils accumulating on plant litter, and how their activities might influence rates of decomposition relative to photodegradation processes.